Welcome back to Anatomiqa. In this session, we will be discussing the details of development of the different structures of the eye. At the end of this session, you should be able to describe the formation of the optic vesicle, lens vesicle, and optic cup. Describe the changes occurring in the associated mesenchyme. Describe development of various structures of eyeball. Describe briefly development of accessory structures of the eyeball. And describe the embryological basis of the congenital anomalies of the eye. Please note this important point. The development of each structure of the eye is described separately, but keep in mind all these events are occurring simultaneously. Only for our orientation we are describing each structure one by one. Before going into actual development of the eye, let us recall the development of the neural tube, which forms the essential part in the development of eye. By the third week of embryonic development, the three definitive primary germ layers are formed in the embryonic plate or the disc. From dorsal to ventral surface of the embryonic disc, they are the ectoderm, mesoderm, and endoderm. Of these three, only ectoderm and mesoderm will take part in the development of ocular structures. We know on the dorsal surface of the embryo, the ectoderm thickens to form the neural plate, which will develop into the neural tube. The neural tube gives rise to the central nervous system, including the ocular structures. Let us recall the formation of the neural tube little bit in brief. To understand how the eye is directly related to the brain, it is important to know the neurulation. Now, over the neural plate, a groove is formed in the center at approximately 18th day of gestation, and the ridges bordering the groove grow into neural folds and starts approximating each other. Just before fusion of the neural tube, an area of cells on the crest of each of the neural folds gets separated from the ectoderm. These are called neural crest cells. They form islands of cells within the mesoderm and surrounds the neural tube. As the neural crest cells are getting separated from the neural folds, the neural fold, which was continuous, now splits into two parts. The two edges ventral to the neural crest cells fuse to form the neural tube, and the ectoderm is now called neuroectoderm. The two edges on the dorsal surface of the neural crest cells fuse with each other and forms the surface ectoderm. Neural and surface ectoderm differ in anatomic location and in differentiation potentials. The cranial end of the neural tube, which is formed, now expands into three dilatations called prosencephalon or forebrain. The prosencephalon further divides into telencephalon, which develops into cerebrum, and diencephalon, which develops into thalamus, hypothalamus, and epithalamus. The second dilatation is the mesencephalon, which gives rise to midbrain, and the third dilatation is the rhombencephalon or hindbrain, which further divides into metencephalon, which develops into pons and cerebellum, and myelencephalon, which develops into medulla oblongata. Diencephalon of forebrain is the focus region in the development of eye. Since eye develops from the diencephalon, which is the point to be remembered, Further development of brain will be discussed in the session on development of nervous system. The neural tube starts closing from the neck region and extends on either sides. The anterior end and the posterior end of the neural tube closes at a later stage. Approximately on day 22, indentations or shallow grooves are formed as evaginations from the wall of diencephalon in the inner surface on both sides even before the tube is completely closed. These indentations are called optic pits or optic grooves. Let us enlarge the region where the optic grooves are formed and study the further development of eye. These are the diagrams of the sections taken at the level of diencephalon and the formation of optic vesicle. Before the fusion of neural folds, the shallow optic grooves are formed at the region of development of diencephalon. As the neural folds are fusing, simultaneously the optic grooves are evaginating from the wall of diencephalon and expands to form the optic vesicle. Once the neural folds are fused, the diencephalon is separated completely from the ectoderm, now called as surface ectoderm. In between the diencephalon and the surface ectoderm, there are neural crest cells and mesoderm. Neural crest cells and mesoderm collectively make up the mesenchyme from which the connective tissue and associated structures of the eyeball and orbit develop. Let us enlarge and focus only on the region where optic vesicle is developing. So approximately on day 25, after the neural tube has closed, the optic pits expand laterally and form sac-shaped extensions or outpocketings called the optic vesicle. The cavity within the optic vesicle is continuous with the lumen of the diencephalon of the neural tube. The surface of each vesicle expands until it comes in contact with the surface ectoderm. Gradually, the two structures become separated from each other by mesoderm and neural crest cells. The optic vesicle induces changes in the surface ectoderm, which is necessary for the development of lens. The influence that one developing structure has on another developing structure is called as induction. Inducing is very much necessary in the formation of the eye. The surface of the optic vesicle induces changes in the surface ectoderm in the formation of lens. In turn, the surface ectoderm induces the optic vesicle to form optic cup. Apart from induction, there are genes named PAX6 and PAX2 which are responsible for the development of the eye. PAX6 is expressed predominantly in the optic cup and lens plecord. PAX2 is expressed predominantly in the optic stalk. Now, how is the optic stalk formed? As the optic vesicle evaginates, the tissue joining the vesicle to the neural tube, that is diencephalon, constricts. This constricted portion between the optic vesicle and the diencephalon is called the optic stalk. The cavity of the optic vesicle, optic stalk, becomes continuous with the cavity of the diencephalon that will become the third ventricle. While the wall of the optic vesicle is in contact with surface ectoderm, it thickens and flattens to form the retinal disc. The lower wall of the optic vesicle and the optic stalk begins to buckle and move inward towards the upper and posterior walls. This invagination forms a cleft called the optic fissure. Optic fissure can also be called as embryonic fissure or fetal fissure or choroidal fissure or the retinal fissure. With this invagination of the lower wall and formation of optic fissure, the optic vesicle, which was a complete ball-shaped structure, now forms two-layered cup-shaped structure called the optic cup. The same invagination can be observed in the transverse section at the level of optic stalk. The invagination occurs at the lower wall of the optic stalk, and observe the two-layered wall which is formed with a cavity inside. The cleft formed by the invagination forms the optic fissure. The mesenchyme, which enters the optic fissure, develops into the hyaloid vessels. The fissure allows the hyaloid artery to reach the inner chamber of the eye. The inferior wall continues to move inward, pulling the anterior wall of the optic vesicle with it, and places the retinal disc in the approximate location of the future retina. Now, the edges of the optic fissure grow towards one another and begin to fuse at 5 weeks. Fusion starts at the center and proceeds anteriorly towards the rim of the optic cup and posteriorly along the optic stalk. Closure is complete at 7 weeks, forming the two layers of the optic cup and optic stalk. The mouth of the optic cup becomes a round opening which forms the future pupil. Here you can observe the optic fissure is completely closed. The cavity between two layers is obliterated and the hyaloid vessels, that is artery and vein, are completely incorporated inside the tissue. Once the optic vesicle is induced by the surface ectoderm, it begins to invaginate and forms the double-walled optic cup. Now, the optic cup has inner layer and outer layer separated by the intraretinal space. The two layers of cells, where both are neuroectodermal in origin, are continuous with each other at the rim of the optic cup. The cells of the inner and outer layers of the optic cup are positioned in such a way the apex of the cells in the inner layer faces the apex of the cells in the outer layer, and both layers of cells are separated by the intraretinal space. Gradually, the two layers approach each other and the intraretinal space becomes potential space and gets obliterated. The outer layer of the optic cup will become the first layer of the retina, the retinal pigment epithelium, the outer pigmented epithelium of the ciliary body, and the anterior iris epithelium. The inner wall become the neural retina, the inner non-pigmented ciliary body epithelium, and the posterior iris epithelium. The surface ectoderm is now induced by the developing optic cup through reciprocal relationship. As the surface ectoderm comes in contact with the optic vesicle, as the invagination of the optic cup is occurring, the surface ectoderm adjacent to the vesicle begins to thicken, forming the lens plate or the lens placode. This thickening is caused by an elongation of the ectodermal cells and by a regional increase in cell division. Cell division stops or ceases in the center of the lens plate, and cell division accelerates or increase in the periphery of the lens plate. Because of this, the lens plate invaginates rapidly, forming a pit called lens pit. The invagination continues and the two edges fuse to form the lens vesicle. The lens vesicle loses contact and then separates from the surface ectoderm approximately during the 5th week. As it loses contact with surface ectoderm, the lens vesicle comes and lies in the mouth of the optic cup. If the area of contact between the optic vesicle and surface ectoderm is less than normal, a perfectly formed but microphthalmic eye can result. Let us now focus only on the lens and see its further development. The lens vesicle is a hollow sphere composed of a single layer of cells. The apical surface of the cells lines the lumen, and the basal aspect is covered by a thin basal lamina or basement membrane. With the addition of more material, the basal lamina will become the lens capsule. Once the lens vesicle is formed, the posterior epithelial cells adjacent to the future vitreous cavity elongate. As the posterior cells elongate, they fill in and obliterate the lumen within the lens vesicle. The developing retina induces the elongation of the posterior epithelial cells. The orientation of the lens is also influenced by the developing vitreous. These elongated posterior epithelial cells lose nuclei and become highly transparent epithelial cells called the primary lens fibers. These primary lens fibers form the embryonic nucleus at the center of the lens. Because the epithelial cells in the posterior surface elongate to form the primary lens fibers in adult lens, there is no epithelial layer beneath the posterior lens capsule. The anterior wall of the lens is lined by simple cuboidal epithelium, which is called lens epithelium. The anterior lens epithelium is active mitotically throughout life. These cells migrate to the equatorial region of lens. These migrated cells elongate anteriorly and posteriorly, forming new lens fibers. The nuclei are arranged in such a manner. If a line were drawn to connect the cellular nuclei within a particular lens fiber layer, an arcuate line is formed. This configuration is called the lens bow. As they elongate, they lose their nuclei and organelles and are filled with cytoskeletal proteins which maintain the conformation and transparency of the lens. These fibers are called secondary lens fibers. The first layer of secondary fibers is completed by 7th week. Secondary lens fibers continue to form and each layer surround the previous layer. Mitosis, cell elongation and lens fiber formation continue throughout development and throughout life. The lens is initially spherical in shape but becomes more ellipsoid with additional fibers. The lens capsule is evident at 5 weeks, evolving from the basement membrane of the invaginating surface ectoderm and from secretions of the lens epithelium. Mesenchyme surrounds the eye primordium from outside and also through the choroid fissure. invades the inside of the optic cup. In the choroid fissure, the mesenchyme forms the hyaloid vessels which supply the lens during intrauterine life. The hyaloid artery forms the vascular layer on the inner surface of the retina and also forms highly branching network that fills the vitreous cavity between the lens and the retina. Near the posterior surface of lens, it forms the vascular network covering the surface of the lens called the posterior tunica vasculosa lentis. By the end of the second month, the hyaloid vasculature is fully formed. To understand further arrangement of the blood vessels, let us observe the lens and optic cup from ventral aspect. Branches near the lens equator anastomose with the annular vessel at the margin of the optic cup. The annular vessel sends loops forward onto the anterior surface of the lens to form the anterior tunica vasculosa lentis during the 7th week. It is the 7th week, not the 2nd week. These networks, blood vessels, supply the developing lens until the aqueous humor is produced. The interstitial spaces of the hyaloid network is filled with mesenchyme, which develops into a transparent gelatinous substance called the vitreous body. Gradually, the hyaloid vessels in this region are obliterated and disappear during fetal life, leaving behind the hyaloid canal. By the 7th month, no blood flow is present in the hyaloid vasculature, which normally should be completely reabsorbed by birth. We have seen the development of optic cup and how it acquires two layers. Now these two layers of optic cup are called the outer layer or deep layer, which is close to the mesenchyme, and the inner layer or the superficial layer, which is close to the developing lens. Further, the outer and inner layer are divided into posterior 4/5 part and anterior 1/5 part. The posterior 4/5 part of the optic cup layers develops into retina, and the anterior 1/5 part gives rise to ciliary body and the iris. So let us see the further development in detail. The retina develops from the walls of the optic cup. In the invaginated optic cup, the outer, deeper, thinner layer becomes the retinal pigment epithelium. The more inner, superficial, thicker layer differentiates into the neural retina. The two layers are separated by an intraretinal space, which is the original cavity of the optic cup. As the inner layer thickens more and more, the two layers of the retina fuse with each other and the intraretinal space obliterates and disappears. Because the optic cup is an outgrowth of the forebrain, the layers of the optic cup are continuous with the wall of the brain. If the retinal pigment epithelium has to develop, which is the first retinal layer to differentiate, it is essential that the two layers of the optic cup oppose each other. Approximately 3 to 4 weeks of development, cellular structures, melanosomes and pigmentation begin to appear in the outer layer. This is the earliest pigmentation evident in the embryo. The cells are cuboidal to columnar in shape, and The base of each cell is directed externally towards the developing choroid, and the apex is directed internally towards the inner layer of the optic cup. Under the influence of the developing lens, the inner layer of the optic cup, close to the retinal pigment epithelium, proliferates to form a thick neuroepithelium. The inner layer, which is close to the vitreous body, develops into nerve fiber layer. The inner layer of optic cup is divided into posterior 4/5 called the pars optica retinae, developing into neural retina, and anterior 1/5 called pars ceca retinae, developing into ciliary body and iris. The pars optica retinae differentiates into 3 layers from outside inwards towards the vitreous body: matrix, mantle, and marginal layers. The matrix layer contains cells bordering the intraretinal space and differentiates into rods and cones. The mantle layer gives rise to neurons and supporting cells, which includes bipolar, horizontal, amacrine, Muller, and ganglion cells. On the surface, the marginal layer is the non-nuclear fibrous layer that contains axons of ganglion cells. Nerve fibers from the ganglion cells converge towards the optic stalk, which develops into the optic nerve. Because of this development, light impulses pass through most layers of the retina before they reach the rods and cones. The fetal or the optic fissure along the optic stalk closes around the hyaloid vessels, which gets buried inside the fused optic fissure. This portion of the hyaloid vessels within the stalk become the central artery and vein of retina. Early in the 4th month of development, primitive retinal vessels emerge from the hyaloid artery near the optic disc and enter the developing nerve fiber layer, which was discussed during hyaloid arterial system. These vessels of the retina continue to develop gradually, forming the arterioles, venules, and capillary beds, and is completed approximately by 3 months after birth. The mesenchyme proliferates and migrates around the optic cup, and once the cells reach their destination, they proliferate and differentiate, contributing to the connective tissue of the eye and orbit. Let us discuss the mesenchymal differentiation and its derivatives one by one. Let us start with outermost layer, the fibrous layer which is formed by the cornea and sclera. The loose undifferentiated mesenchyme anteriorly is differentiated into 2 layers, inner and outer layers, by the formation of the anterior chamber. Inner layer, which is present in front of the lens and iris, is called the iridopupillary membrane, and an outer layer present in front of the anterior chamber. This layer gives rise to the cornea. Let us enlarge the anterior part and look at the development of the cornea. The layers of cornea are formed by surface ectoderm and the mesoderm. The surface ectoderm gives rise to anterior epithelium of the cornea. The outer layer of the mesoderm in front of the anterior chamber gives rise to thick middle layer, the substantia propria or stroma, which is continuous with the sclera posteriorly. The mesoderm also gives rise to Bowman's layer or the Bowman's membrane just deep to anterior epithelium, Descemet's layer or the Descemet's membrane, and corneal endothelium which borders the anterior chamber. At the end of the 5th week, the optic cup, now called eye primordium, is completely surrounded by loose, undifferentiated mesenchyme. This tissue soon differentiates into an inner layer comparable with the pia mater of the brain and an outer layer comparable with the dura mater. The outer layer develops into the sclera and is continuous with the dura mater around the optic nerve. The sclera first starts developing anteriorly from condensations in the mesenchyme near the junction of developing sclera and cornea, which is called limbus. Growth continues posteriorly until the sclera reaches the optic nerve, and by the end of the 3rd month, the sclera has surrounded the developing choroid from the inner layer. Coming to the middle layer of the eyeball, uvea, it includes choroid, ciliary body and iris. The inner layer of the surrounding mesenchyme develops into the highly vascularized pigmented layer, the choroid. The mesenchyme that forms the choriocapillaries must be in contact with the developing pigment epithelium to differentiate. Let us enlarge the anterior part of the developing eye and look at the development of ciliary body. The region of the outer layer of the optic cup develops into the outer pigmented epithelium of the ciliary body. This layer begins to form ridges late in the 3rd month. We have seen previously that the anterior 1/5 of the inner layer of the optic cup, the pars ceca retinae, remains one cell layer thick and further divides into 2 parts, pars ciliaris retinae and pars iridica retinae. The inner layer of the optic cup, that is pars ciliaris retinae, develop into non-pigmented epithelium of the ciliary body. This non-pigmented epithelium also grows and folds with outer layer. These folds, almost 70 in number, become the ciliary processes. Neural crest cells differentiate into stromal elements. By 14th week, the fenestrations in the capillaries present in the processes are visible. Now let us further enlarge only the pars ceca retinae and see the further development. The pars ciliaris retinae is easily recognized by its marked folding called ciliary processes. Externally, the ciliary process is covered by a layer of mesenchyme that develops into the ciliary muscle. On the inner surface, it is connected to the lens by a network of elastic fibers called the suspensory ligament of lens or zonula. Contraction of the ciliary muscle changes tension in the ligament and controls the curvature of the lens. Now coming to the development of the iris, by the end of the 3rd month, the lip or anterior end of the optic cup begins to elongate and grows between the lens and the differentiating mesenchyme going to form the cornea. The outer layer of the optic cup, which continues in front of lens, develops into anterior epithelium of the iris. The pars iridica retinae, which is a part of the anterior 1/5 or inner layer of the optic cup called persica retinae, forms the posterior epithelium of iris. These layers remain separated from each other for some time by a space called the marginal sinus. Simultaneously, the region between the optic cup and the overlying surface epithelium is filled with loose mesenchyme. The underlying ectoderm of the optic cup develop into the sphincter pupillae and the dilator pupillae muscles in the mesoderm. The layers of the iris in the adult from posterior to the anterior is the pigment containing posterior epithelium of iris, the unpigmented anterior epithelium of iris, and a layer of richly vascularized connective tissue containing the sphincter and dilator pupillary muscles. Now let us see what are the layers which are differentiated from the two layers of the optic cup. The outer layer of optic cup in the posterior part differentiates into pigment epithelium of retina, which is the first layer of retina, and in the anterior part into pigmented epithelium of ciliary body and the anterior epithelium of iris. The inner layer of optic cup in the posterior 4/5, which is called pars optica retinae, differentiates into neural retina, which gives rise to the rest 9 layers of the retina, and anterior 1/5 of inner layer called persica retina differentiates into non-pigmented epithelium of ciliary body and posterior epithelium of iris. So coming to the pupillary membrane or iridopupillary membrane, it lies ventral to the lens. The iridopupillary membrane is derived from the mesenchyme present between the lens and the anterior chamber of the eye. It is composed of mesenchyme and network of vessels derived from anterior ciliary arteries. The pupillary membrane undergoes regression by macrophage phagocytosis in the 6th month and disappears completely by the 8th month, leaving a space between the anterior margin of iris which forms the pupil. The light now can pass directly through the pupil without interruption to the lens. Now let us discuss the development of chambers of the eye. The mesenchyme in front of the lens undergoes vacuolization to form anterior chamber of the eye. A mass of cells of neural crest origin and mesenchyme accumulates in the angle of anterior chamber adjacent to the ciliary body and the root of iris. These cells develop into the epithelium lining the trabecular meshwork. The endothelium is now continuous, lining the anterior chamber, trabecular meshwork, and the iridocorneal angle. The anterior chamber formed through vacuolization splits the mesenchyme into 2 layers. An inner layer in front of the lens and iris called the iridopupillary membrane and an outer layer continuous with the sclera, which gives rise to substantia propria of the cornea and corneal endothelium. The space present between the iris anteriorly and the lens and ciliary body posteriorly develops into the posterior chamber. Through the development, the pupillary membrane disintegrates and disappears. With the disintegration and disappearance of the pupillary membrane, a space is formed at the margin of the iris called the pupil. Now, the posterior chamber communicates with the anterior chamber through the pupil and are filled with fluid called the aqueous humor produced by the ciliary process of the ciliary body. The clear aqueous humor circulates from the ciliary body to the posterior chamber and then into the anterior chamber, providing nutrients for the avascular cornea and lens. From the anterior chamber, the fluid passes through the scleral venous sinus, also called as canal of Schlemm, at the iridocorneal angle, where it is resorbed into the bloodstream. Blockage of the flow of fluid at the canal of Schlemm is one cause of glaucoma. The presence of the developing lens is essential for normal accumulation of vitreous. The mesenchyme invades the optic cup through the retinal or optic fissure. The mesenchyme becomes entrapped as the retinal fissures close and seal off the optic cup. This entrapped mesenchyme, along with the cells originating in the ectoderm, forms the primary vitreous body. The primary vitreous body loses its vasculature and cells, transforms itself into an acellular gel known as the secondary vitreous body. The hyaloid artery regresses and forms a vestigial structure which remains in the adult eye as the hyaloid canal. Now, coming to the development of the optic nerve, the optic stalk joins the optic vesicle to the diencephalon of the forebrain. As the optic fissure develops through invagination along the inferior wall of the optic stalk, the two-layered wall is created, the inner and the outer layer with a space in between. The optic fissure is invaded by the mesenchyme, which gives rise to hyaloid vessels. The outer layer of the optic stalk becomes the neuroglial sheath that surrounds the optic nerve. It also gives rise to the glial components of the lamina cribrosa. The nerve fibers of the ganglion cells of the retina returning to the brain pass through the inner wall of the optic stalk. As a result, the continuously increasing number of nerve fibers, the inner wall of the optic stalk grows and the inner and outer walls of the optic stalk fuse. During the 7th week, the choroid fissure closes and a narrow tunnel is formed inside the optic stalk, which is obliterated later by the fusion of inner and outer walls. Cells of the inner layer provide a network of neuroglia that support the optic nerve fibers. The optic stalk is thus transformed into the optic nerve. Its center contains mesenchyme with a portion of hyaloid vessels, later called the central artery and vein of the retina. Optic nerve is covered by pia and arachnoid matter, which forms the continuation of choroid, and dura mater, which is the continuation of sclera. Because it develops as the continuation of diencephalon, it becomes a part of the brain. It is not completely myelinated until 3 months after birth. It is myelinated by oligodendrocytes and is not capable of regeneration after transection. We are now left with development of ocular associated structures, which includes eyelids, orbit, and extraocular muscles. To start with eyelids, early in the 2nd month of gestation, surface ectoderm forms folds which are filled with mesenchyme. They begin to grow towards one another anterior to the developing cornea. These folds will develop into eyelids. The upper fold arises from the frontonasal process and the lower fold arises from the maxillary process. The eyelid margins meet and fuse during the 3rd month of development, and they remain fused until all the structures of the eyelid are developed. The layer of ectoderm which covers the anterior surface becomes the epidermis, and the layer which lines the inner surface becomes the palpebral conjunctiva. The ectoderm which covers the anterior surface of cornea and sclera develops into orbital conjunctiva. The junction between the orbital and palpebral conjunctiva forms the superior fornix and inferior fornix related to upper eyelid and lower eyelid respectively. Coming to the last part of development, the connective tissue and extraocular muscles. All the extraocular muscles of the eyeball develop from mesenchyme. The connective tissue components are derived from neural crest. The muscles innervated by cranial nerve 3, that is the oculomotor nerve, are derived from the first pair of somites. The lateral rectus muscle, innervated by the 6th cranial nerve or abducent nerve, develops from the mesenchyme of the maxillomandibular area. The superior oblique muscle, innervated by the 4th cranial nerve or trochlear nerve, is derived from the 2nd pair of somites. The newborn exhibits poorly coordinated eye movements during the early years, indicating that the extraocular muscles are not fully developed. Nasolacrimal system includes the lacrimal gland and the nasolacrimal drainage system. The main lacrimal gland develops from epithelial buds that arise from the temporal portion of the conjunctiva of the superior fornix. The conjunctiva is ectodermal in origin, so the lacrimal gland also will be ectodermal in origin. The lacrimal gland continues to develop after birth up to the age of 3 to 4 years. The nasolacrimal drainage system develops from a cord of surface ectodermal cells that becomes buried below the maxillary mesenchyme. This cord fragments and form the canaliculi, the nasolacrimal duct, and the lacrimal sac. This part was discussed in the development of face. Let us recall once again how it is developed. Initially, the maxillary prominences and the lateral nasal prominences are separated by a deep furrow or cleft called the nasolacrimal groove. So, these are nasolacrimal grooves forming between the lateral nasal processes and the maxillary processes as the development proceeds. Let us enlarge the nasolacrimal groove and see its further development. In the floor of the nasolacrimal groove, the ectodermal cells first proliferates to form a solid epithelial cord of cells. This epithelial cord then detaches from the overlying ectoderm. When we take a section across and look at it, this is how it is seen from one end. Now the epithelial cord of cells in the center disappears due to apoptosis. That is the programmed cell death, and a canal is formed which is called as the nasolacrimal duct. The cranial or upper end of the nasolacrimal duct widens to form the lacrimal sac. Following detachment of the cord from the groove, the maxillary and lateral nasal prominences merge with each other. The nasolacrimal duct then runs from the medial angle of the eye to the inferior meatus of the nasal cavity. The nasolacrimal duct usually becomes completely canalized only after birth. The tables show the embryologic derivation of ocular structures. You can pause the video and go through the derivatives. Coming to the congenital anomalies or abnormal development of the eye, starting with persistent pupillary membrane. Remnants of the central portion of the pupillary membrane that do not reabsorb may be seen with a biomicroscope and appear similar to strands of a spider web attached to the surface of the iris. A persistent pupillary membrane may have a variety of presentations, from a single strand of connective tissue anchored at one or both ends to several interconnecting strands. Pigment cells also might be incorporated. A persistent pupillary membrane is present in 17% to 32% of the population. Coloboma iridis is a cleft in the iris caused by failure of the choroid fissure to close in week 7 of development and may extend into the ciliary body or the retina or the choroid or the optic nerve. A palpebral coloboma, a notch in the eyelid, results from a defect in the developing eyelid. It can occur either during fusion at the 3rd and 4th months of embryologic development or during reseparation, which occurs in the 6th or 7th months. Congenital cataracts are opacities of the lens and are usually bilateral. They are fairly common and may result from the following: rubella virus infection, toxoplasmosis, congenital syphilis, Down syndrome or trisomy 21, and galactosemia, which is an inborn error of metabolism. Congenital glaucoma, also called as buphtalmos, is increased intraocular pressure due to abnormal development of the canal of Schlemm or the iridocorneal filtration angle. It is usually genetically determined but may result from maternal rubella infection. Observe the bulging of the eyeball in these pictures. The site of retinal detachment is between the outer and inner layers of optica. That is between the retinal pigment epithelial layer and the outer segment layer of rods and cones of the neural retina. Microphthalmia is a small eye usually associated with intrauterine infections from the torch group of microorganisms, that is the toxoplasma, rubella virus, cytomegalovirus and herpes simplex virus. Together these are called as the torch group of microorganisms. Another cause for the microphthalmia can be minimal contact between the surface ectoderm and optic vesicle. Anophthalmia is the absence of the eye. It is due to failure of the optic vesicle to form. Papilledema is the edema of the optic disc or the papilla due to increased intracranial pressure. This pressure is reflected into the subarachnoid space which surrounds the optic nerve. Retinitis pigmentosa is a hereditary degeneration and atrophy of the retina. Retinitis pigmentosa may be transmitted as an autosomal recessive, autosomal dominant or X-linked trait. Cryptophthalmos is a condition of total absence of eyelids and the skin of forehead is continuous with that of the cheek, in which the eyeball is completely submerged beneath the skin. In the fourth picture, you can see the bulged eyeballs beneath the skin. With this, we have completed the development of eye. Thank you for watching till the end. If you find the video useful and helped you in understanding the subject, please like, share and subscribe to the channel. Thank you.